Amber asked me to talk a little bit today about the work that we're doing in collaboration with SLC6A1 Connect and the nonprofit that I work for, Combined Brain. This is going to be switching gears completely from the previous presentation, but we'll overlap a little bit in terms of showing some of the basic research resources that we have available for both basic researchers and clinical researchers studying SLC6A1. So as I look around the room, I don't probably need to introduce a whole lot about our organization, but Combined Brain, the mission for Combined Brain and our goal is really kind of a lofty goal, but it's to it's a consortium of about 60 different, actually probably now 75 different patient advocacy groups, most of which are rare monogenetic neurodevelopmental disorders. And our goal is really to expedite the development of treatments, whatever form that takes, by collaborating across disciplines with scientists, clinicians, and various industry members. So if you look, I have no idea if this pointer works. Is there a pointer? Oh, there is. So this is a really outdated logo for us, but if you look, there we go. SLC6A1 has been a member since we launched Combined Brain and has been really heavily involved in a lot of the projects or the initiatives that we've spearheaded in the last 2 years. So I think when I landed last night, I realized that I think I show this slide literally every time I come to a conference here. So I won't belabor it, but I like it because it really shows a pretty graphical representation of what it looks like for the pipeline of developing the drug development process. So on the left-hand side, you identify the causal gene for the disorder, and then one of usually the first next steps is develop animal models and cell models for basic researchers to study and interrogate that pathogenic mechanism. And then also a critical component is to establish a natural history for the disorder to see how it changes with age. Additional investigations into the molecular consequences of that pathogenic gene. As you go further to the right, you get more into more in-depth clinical profiling. So on the top here, you say the importance of a disease concept study, which probably Dr. Goodspeed will talk about a little bit more later, but I'll just introduce it. And then, of course, the identification of biomarkers, so objective measures that can tell you whether the disease is progressing or otherwise just categorize the disease stage. And then one of the last things is really just to start developing and focusing and validating patient-centered outcome measures, which also Dr. Bichelle will speak about towards the end of the day today. And then clinical trials or pilot clinical trials. And those, I think, will also be addressed in the next couple talks. So what I want to do is spend my last 11 and a half minutes talking about the areas along this pipeline that we have really collaborated with SLC6A1 to see how we can advance the field for both basic researchers as well as the clinical researchers in the room. So the first would be the cell models that we have collaborated with the SLC6A1 community to establish. And this is certainly not all of the cell models. I know I can see faces of people who have their own individual patient-derived cell models in their own labs, but one of the first things that we did as an organization was to launch a patient-governed biorepository. So these are samples that are collected at the request of the foundations, and we hold them and we distribute them to researchers with considerably less administrative red tape, and we can distribute them pretty quickly and easily. So we have so far we have established 2 iPSC lines, which are kept at our commercial facility. And then we have 3 primary cell lines from additional patients, as well as a sibling control that are in our repository and readily available to any researcher who requests them. one note that I wanted to remind myself here is that if you are an academic researcher and for a variety of reasons would like to transfer in existing cell models, whether they're fibroblasts, neurons, iPSCs, other primary cells, that's something that's very easy for us to do. And all you would need to do is just send me an email or get a hold of me somehow, and we can do that pretty easily. Okay, so the second thing that I'll introduce just really briefly is natural history, the importance of natural history data, which I don't really need to belabor for anybody in the room here. And there are several efforts on this front right now for 66A1. And so what I'll say is just a really small component of that is that we at Combined Brain have really collaborated with an online natural history, a data collection platform called the Matrix. And through that platform, there are a variety of different, typically caregiver completed assessments. You can see them outlined here. We have the ClinGen surveys that do a pretty in-depth medical history as well as developmental assessments. And then in addition to ClinGen surveys, we have everybody's favorite, the Vineland Adaptive Behavior Scale. And then we also have the PELS Seizure Survey, which is just recently generated by Dr. Grinspan. Is Zach here? No, I don't see Zach. Okay. So we have a seizure survey as well on this natural history platform. And we have quite a few registrants. We have 17 66A1 individuals who've registered. Most of them have completed more than one survey, and 8 have completed all surveys, which is kind of a beast to complete. One thing that I want to point out that is important about using this platform as an organization is that any survey that the participant, a caregiver, completes can download it and share it to whoever needs that information. So if there's another clinical researcher in the room who needs a Vineland completed for whatever their ongoing projects are right now, the individual participant can download it and share it to the individual researcher whenever they choose. So there are no restrictions on data sharing. It's owned by the individual participant as well as 66A1. Okay, so the next thing that I probably will focus the most on is the need and the role for biomarker discovery along this sort of therapeutic pipeline. This is probably the one that's the most nearest and dearest to my heart and has been one of the major pushes for things to collect for our biorepository. So this is probably the world's most hideous table right here, but I put it on here just to give you an idea of how active the patient community has been in response to a call to start collecting these types of samples. So what you can see is that we have 11, so we've started collecting a variety of biofluids. So we collect like, for example, when we fly to conferences like this one or other family conferences, we collect plasma, we collect dried blood spots, we collect whole blood, and we also collect urine. So for SLC6A1, so far we have collected 11 plasma samples, 11 dried blood spots, 9 whole blood samples, and 7 urine samples. I put this really ugly table on here so just so you can sort of get an idea of the types of variants and individuals that we're collecting from. And you can see that we collect from unaffected, we try and make it age-matched control subjects, which tend to be siblings. And then in addition to the biofluid or biological sample that we collect, we again have them complete those same surveys on the Matrix platform so that the biological samples, anything that you receive, will have this variety of clinical phenotypic information to go with it. Okay, so I'll just touch really briefly on disease concept studies, but I think Dr. Goodsby will probably talk about this a little bit more in depth later today, but briefly, a disease concept study, it's a really great way to gauge the symptoms that are most burdensome to families, caregivers, patients themselves. So you do that. It's a semi-qualitative study with interviews by caregivers, clinicians, educators who are familiar with the particular disorder, in this case 66A1. And you interview experts in whichever fashion they are here, until you see no additional symptoms being reported in your interviews. And then the goal is that you publish this and you put it in the scientific literature. And in fact, this is what Kim, Dr. Goodspeed, and Dr. Bichelle have done just recently. In fact, published a draft conceptual model for 66A1 this past year. This is just an example of the types of symptoms that are coming out of this. But one of the primary reasons to do this is it really shapes the outcome measures that you're going to include in your pilot clinical trials or any other future clinical trials based upon the symptoms that are reported by these clinical experts or obviously caregivers. And interesting, one of the biggest concepts that came out of this study is a need for improved communication measures between caregivers and their kids. So actually this leads really nicely into the next part on this schematic, which is developing patient-centered outcome measures. And so one really perfect example of this is one of the main initiatives and projects that we've started as an organization is to partner with a team at Duke, Dr. Christy Ziegler and Bryce Reeve, who developed an observer-reported communication ability measure. This is short for the ORCA. And they developed it based on Angelman syndrome. And we recently received a grant from the FDA to expand the ORCA to 12 different other neurodevelopmental disorders. And we're currently working on expanding this again past those initial 12 to include groups like 6A1. And this is a really important a study, as it would be a validated outcome measure for changes in communication, which caregivers already told us was one of the main priorities and most burdensome symptom to families. And then you can present this to the FDA as a potential outcome measure for pilot clinical trials, which you initiate. Okay. I think that was all. Okay. I think I included this last one here just to give an appreciation for the fact that 66A1 Connect and the foundation as a whole has really gotten a great appreciation for how investment and community involvement across this pipeline is really critical for the development of clinical trials on the very far hand right of the schematic. Okay, so this is the team. We're led by our fearless leader, Dr. Terry Jo Bichelle, which I feel like will speak probably later, and a lot of other people who have done the vast majority of the work that I just presented here today. And if you have any questions or if you have any resources that you'd like to contribute to Combined Brain, this is my email address. And thank you very much for your time.